Yeah, thank you again for the opportunity to present my work here. It's a paper optimizing the PBS1 decoy system to confer resistance to potivirus infection in Arabidopsis and soybean that was published in MPMI of July last year. So plants have several ways in which they protect themselves from pathogens, but the one that my work focuses on is effector-triggered immunity, in which pathogens like this bacteria or fungi secrete into the cell as part of their infection cycle effectors. These effectors are recognized by the plant through their effect on Nla proteins or nucleotide binding leucine-rich repeat proteins. Whether this recognition is direct via the NLR itself or indirect through its guarding of another protein, once the recognition occurs, these NLRs signal for a whole suite of immune responses, a characteristic one being the hypersensitive response or the controlled death of the cell that starves these pathogens of an environment in which to live and leach nutrients from, and therefore provides immunity to the pathogen. And so this sort of genetic disease resistance represents a really cost-effective and environmentally friendly way to protect our crops from pathogens in the field. However, the use of resistance proteins like this is hampered, firstly by the lack of proteins detecting specific pathogens in existing germplasm, as crops are normally quite bottlenecked, and as well as the lack of R protein characterization, because even though new R proteins are being characterized on a regular basis, for any given pathogen or pathogen strain that's an issue in the field, it is somewhat unlikely that there'll be a characterized R protein in a species of plant that's able to be crossed into that crop in an effective and timely manner. And that's where the PBS1 decoy system comes in. So the PBS1 decoy system is predicated on PBS1. It's a protein kinase that is guarded by the NLR RPS5 in Arabidopsis. And together they provide immunity to Pseudomonas syringae. As part of its infection cycle, Pseudomonas enters into the plant cell a whole suite of effectors, including one called AvrPphB. And AvrPphB is a protease that targets a lot of proteins that look like PBS1 and cleaves them to try and prevent their functioning in the immune response. So when PBS1 is cleaved, it produces a conformational change that is somehow sensed by RPS5. RPS5 is then predicted to undergo its own conformational change and then somehow signal for that hypersensitive response, and that controlled cell death produces resistance to Pseudomonas syringae in a manner that is so effective that disease symptoms don't develop and these plants are completely healthy. So I'll talk a little bit more about that PBS1 conformational change and use this diagram of PBS1 that's based on the structure of BIC1, a somewhat similar protein. And you can kind of see that along this line, the protein is divided into two that is just linked by this cleavage loop, which is a more accurate representation of this loop here. And so when this is cleaved, it produces enough conformational dynamic shift in the protein that this is sensed by RPS5, possibly through its interaction with this SCMPH motif. So because RPS5 is activated by a conformational change in PBS1 rather than the cleavage itself, or the effector, any protein capable of cleaving PBS1 and causing that conformational change should be able to activate RPS5 and confer resistance. And so in this work, the activation loop of PBS1 was mutated to contain the consensus cleavage site for the tenant mosaic virus Nla Protease. So instead of AvrPphB cleaving PBS1, the tenant mosaic virus Nla Protease is cleaving PBS1 TUNV, which is guarded by RPS5 in exactly the same way and should produce resistance. At least that's the idea. We chose the tenant mosaic virus because it infects a lot of Brassicaceae that we like to eat, and we chose the Nla Protease because it is integral to the way that the tenant mosaic virus propagates. Tenant mosaic virus produces all of its cellular proteins as one long polyprotein, as you can see across the bottom, and it cleaves that at specific sites to create the actually useful proteins that it uses as part of its life cycle. And all of the sites marked with arrows are cleaved by the Nla Protease. Therefore, using the consensus cleavage sequence for the Nla Protease in PBS1 provides a pretty robust immunity, as it's unlikely that complementary mutations would co-occur both in the Nla Protease and in all of the cleavage sites that it is required to do to allow the tenant mosaic virus to propagate. So we made our PBS1 TUMV protein and expressed it in benthy along with the tenant mosaic virus protease. And you can see that it does cleave. There's a strong cleavage product here that doesn't happen with the wild type PBS1. And this is also followed through in an HR experiment in which RPS5, PBS1 TUMV, Nla Protease, and PBS1 TUMV expressed together in a benthy leaf causes this lesion of dead cells, which is indicative of HR. And of course, in a natural circumstance, this would be happening on a single cell level, but in these HR experiments, we make them happen on a much wider scale so that it's easier for us to visualize. So because this all looked really great, we went ahead and made PBS1 TUMV stable lines in Arabidopsis that were expressing the PBS1 TUMV under a native PBS1 promoter. And we challenged these lines using Agrobacterium mediated delivery of the tenant mosaic virus. So for this, you infiltrate young Agrobacterium or young Arabidopsis plants with Agrobacterium that will deliver DNA encoding for the tenant mosaic virus that had a GFP tagged protein. That DNA is then expressed by the plant and the tenant mosaic virus is assembled and its infection cycle continues naturally, bringing with it that GFP marker. So then when you come to assay the plants for infection, it can easily be visualized under UV light as the blue-green fluorescence from the GFP. And this stands out really nicely against the red-pink of the chloroplast altered fluorescence. So this is what our lines actually looked like. At 11 days post-infection, our wild type Columbia line has a bunch of virally derived GFP in it under condition that has shown us that it's infected with virus, whereas our PBS1 TUMV expressing line has a lot less virus in it. This is really great, but if you leave the plants for an extra week, it begins to look not quite so great. Our PBS1 TUMV line is looking pretty sickly, and under UV light, the white fluorescence here is indicative of dead cells. So even though the immune response is happening, this should have shows that it's happening too slowly to contain the spread of infection, so we get this trailing necrosis. In other words, by the time the cell realizes that it's infected with the virus and starts that HR, that cell death response, it's already infected its neighboring cells. And obviously this is not ideal for a technology that's supposed to prevent the plant from becoming infected and keep it healthy. So we theorized this could be due to a positional mismatch within the cell between the PBS1 and RPS5 system at the plasma membrane and the tenant mosaic virus Nla Protease. AvrPphB, the effector that the PBS1 RPS5 complex normally recognizes, is also tethered to the plasma membrane, whereas the tenant mosaic virus replicates at these viral factories that are made from vesicles co-opted from host endomembranes in the endoplasmic reticulum and the nucleus of the cell. So it might just be that for the PBS1 TUMV protein to come into contact with the TUMV Nla protease, there needs to be so much of it in the cell that it's already infected its neighboring. And so we thought maybe we can change the localization of PBS1 and RPS5. And we knew a little bit about RPS5 localization already. We know that it had acylation motifs that function additively to keep it tethered to the plasma membrane. So this is a wild type RPS5 that's pretty plasma membrane localized. However, when you mutate the myristoylation motifs at glycines 2 and 3, it produces a more cytoplasmic localization that then becomes completely cytoplasmic if the palmitoylation motif at cysteine 4 is also mutated. And this follows through for HR tests as well, in which a mutation in either the myristoylation or the palmitoylation motifs is still tolerated by RPS5 and it can still produce that cell death response. However, when they're both mutated, RPS5 is no longer functional. And that is why when I relocalized RPS5 to the cytoplasm, I'm only using the G23A mutant in the hopes that it would still be effective. So yeah, I wanted to relocalize both PBS1 and RPS5 to the cytoplasm, the nucleus, and to these viral vesicles. And I did this by replacing those acylation sites both in RPS5 and similar ones in PBS1 with either alanines, just to remove their myristoylation motifs to create cytoplasmic constructs, with a nuclear localization signal, and for which I just replaced these 7 amino acids, or I replaced them with the tenant mosaic virus 6K2 protein. And I made this conjugate because it was known that 6K2 forms these viral vesicles. They they produce these blobby puncta that are made from host endomembranes, and these are associated with all of the transcription machinery that's needed to create new virus. And so the thought was that by relocating PBS1 and RPS5 to the 6K2 vesicles, that they would be in like the epicenter of where new virus is being created and should come into contact with it really speedily. So what do my relocalizations actually look like? I'm going to show all of the PBS1 TUMV across the top and all of the RPS5s across the bottom. And these are just the wild type or plasma membrane localized proteins in which they look pretty plasma membrane localized, although you do see some overexpression as a result, or some cytoplasmic streaming as a result of the overexpression. However, for the nuclear localized, you can see that I see strong nuclear association as well as some protein still in the cytoplasm. And this is sort of the opposite is true for the cytoplasmic constructs, in which, especially for PBS1, I still see some nuclear localization, but also a good cytoplasmic accumulation of the protein. For the 6K2 conjugates, I was able to recapitulate that blobby puncta that 6K2 makes on its own, as well as I saw this specific association with chloroplasts, specifically for the 6K2 PBS1 TUMV. And if you zoom into what that actually looks like, all of the chloroplasts in the cell are sort of aggregated together, and inside them, these replication vesicles form. And this has sort of been previously shown as something that the tenant mosaic virus is capable of doing. And these are just Western blots to show that these localization patterns are the result of full-length protein being expressed and not free GFP. So I knew that I could relocalize PBS1 and RPS5. I wanted to know whether relocalized PBS1 TUMV could still be cleaved. And so this is plasma membrane localized PBS1 TUMV, and in the presence of the Nla Protease, we see a cleavage product, which is as we expect. And this is also the case for all of the relocalized constructs. The nuclear localized PBS1 TUMV's cleavage product was a little bit difficult to see, but it is there. This sort of represents a larger trend that I saw for the nuclear localized constructs to be more patchily expressed and just at a lower expression level in general. But yeah, I saw that PBS1 TUMV could still be cleaved at its new localization, and I wanted to know whether the relocalized RPS5 could still function. And for this, I had two tests. The first of which was just a regular HR test in which we're expressing, in this case, plasma membrane localized RPS5, PBS1 TUMV with the tenant mosaic virus Nla Protease, and we can see this lesion of dead cells indicative of the HR response and the immune system functioning as we expect it to. And this is dependent on the presence of RPS5, as it doesn't happen when RPS5 is not being expressed. However, I was concerned that possibly relocalizing RPS5 and PBS1 TUMV would disrupt their association, like maybe the relocalization is somewhat different for the two proteins. And to get around that, I also was using an autoactive version of RPS5 that constitutively signals for cell death, even in the absence of the tenant mosaic virus Nla Protease, or indeed in the absence of PBS1 TUMV. And so you can also see that this produces a lesion of dead cells. However, when we look at my relocalized constructs, the nuclear localized PBS1 TUMV co-expressed with nuclear localized RPS5 and the Nla Protease shows no cell death. And the autoactive version of the nuclear localized RPS5 also shows no cell death. I was also expressing the relocalized PBS1 TUMV constructs with wild type plasma membrane localized RPS5 just to sort of as a control, but I ended up actually seeing cell death with the relocalized 6K2 PBS1 TUMV and the plasma membrane localized RPS5. This was the only combination of constructs in which I did see cell death outside of my positive controls. And I think really shows that RPS5 requires something in the plasma membrane to function. Because these looked good, I went ahead and created stable lines. And yeah, this is just a Western blot showing that the lack of cell death that I see here for my relocalized constructs is not due to lack of protein expression. It really looks like RPS5 requires something about the plasma membrane. So I made these 6K2 PBS1 TUMV stable lines and I challenged them with tenant mosaic virus infection. However, they do not seem to be resistant. These lines are expressing 6K2 PBS1 TUMV in a PBS1 knockout background, just in case having wild-type PBS1 and PBS1 TUMV competing for RPS5 association reduced the efficiency of the immune response. And that may be the case, but the immune response seems to be completely absent, as you see just as much virally derived GFP as we do in our susceptible control. And this is an RPS5 knockout background, as without RPS5 to signal for the hypersensitive response, they should be unable to mount an immune response and be completely susceptible to the virus, which is what we see here, as there's a lot of virally derived GFP. And again, this is backed up by immunoblot, showing that even though there is a huge amount in some lines of PBS1 TUMV being expressed, we still see that accumulation of virally derived GFP. So this raises really interesting questions about RPS5 signaling and why it might require a plasma membrane localization. It's predicted to oligomerize upon activation, but other than this, not much is known about how it might signal for HR. In the light of the recent ZAR1 paper showing that it's a calcium channel, or it can function as one, it would be really interesting to speculate on whether RPS5 might function in a similar way. However, the CC domains of RPS5 are really quite different from that of ZAR1. RPS5 has these acylation motifs that are completely absent from ZAR1, and furthermore, the residues in ZAR1 that were identified as being really necessary for the CC domain oligomerization and association with the plasma membrane are completely absent from RPS5. The CC domain of RPS5, in fact, maps really poorly to the CC domain of ZAR1 structurally, and all that taken together sort of suggests that RPS5 has a different method of signaling, possibly one involving a signaling partner. However, none have been identified previously using mutational screens. All of this could be due either to redundancy or even lethality of those signaling partners when absent. And so I'm currently using a proximity labeling approach using the mini turbo BIRA variant to do a biotinylation bio-ID of possible partners for RPS5. And this raises cool new questions. However, it doesn't help this original issue, which is how to prevent this trailing necrosis in our PBS1 TUMV lines. And so relocalization didn't work. We thought maybe overexpression would. So instead of using the native PBS1 promoter, we created overexpression PBS1 TUMV lines in the hopes that just having a higher amount of PBS1 TUMV in the cell would allow it to come into contact with the Nla Protease in a more timely manner and then prevent this trailing necrosis. And fortunately, this was the case. You see, these are our original lines that have a PBS1 TUMV under the PBS1 native promoter. And you can see that I was able to recapitulate that trailing necrosis phenotype. However, in the overexpression lines, these plants look completely healthy. I think it's interesting to note that the 3 leaves that were originally inoculated all died, and that I saw this in all of my replicates. But that death of those leaves seemed to have sort of saved the rest of the plant, and it's completely free of viral infection. This is also true in the PBS1 knockout background that, again, I made to make sure that wild-type PBS1 and PBS1 TUMV competing for RPS5 association wouldn't slow the immune response. However, because these ones in a wild-type background look so healthy, I think that's not an issue. The only line that I did see viral accumulation in is our susceptible control, the RPS5 knockout background, as without RPS5, it shouldn't be able to mount an immune response. And that is what we see, that there's a huge amount of virally derived GFP here. And this is backed up by immunoblot, in which the lines that are expressing PBS1 TUMV in the presence of RPS5 show no virally derived GFP. And you can also get an idea of the difference in expression level between the native promoter and the overexpression constructs. The expression of PBS1 in the RPS5 knockouts looks like it's lower, but I think this is actually due to the turnover of PBS1 that happens once it's been cleaved, which is happening continuously in these lines as there's ternate mosaic virus producing its Nla Protease. Yeah, so we were also working with Claire Castillo's lab and a postdoc at the time, Aurelie Back, to see whether or not our stable lines could be resistant to ternate mosaic virus when vectored more naturally via aphids. And to do these experiments, plants that are already infected with the tenant mosaic virus that are expressing, again, this GFP tagged 6K2. These have aphids on them that are allowed to feed for 5 minutes and take up the virus. These aphids are then transplanted to uninfected plants and allowed to feed for 24 hours before being removed, and these plants are left for 10 days to develop infection and then assayed for symptoms. And really encouragingly, the results of this look almost identical to when the tenant mosaic virus was affected by Agrobacterium. So again, the lines that are expressing PBS1 TUMV in the presence of RPS5 show little to no buildup of the tenant mosaic virus derived coat protein. The only lines in which we do see that are the wild type control that is not expressing PBS1 TUMV and the RPS5 knockout, in which there's no RPS5, so there's no immune response. So this was a really exciting result for us. It showed that the PBS1 decoy system is able to confer complete immunity to a viral pathogen vectored both by Agrobacterium and more naturally by aphids. But to be really useful, this immunity has to be additive. If we're just replacing the immunity to Pseudomonas syringae with the immunity to tenant mosaic virus, it is sort of a zero-sum game. So to test this, I challenged my stable lines with Pseudomonas syringae expressing either an empty vector or AvrPphB. And so this is the amount of leaves that were infected and the number that actually showed cell death or showed an immune response. And in the empty vector, this is pretty low. However, in leaves challenged with Pseudomonas syringae that are expressing AvrPphB, all of the lines that are expressing a wild type PBS1 as well as PBS1 TUMV showed almost complete recognition of AvrPphB, which is great news. It's also interesting to note that the lines in a PBS1 knockout background that are only expressing PBS1 TUMV showed an intermediate level of AvrPphB recognition. It's probably indicative of the fact that AvrPphB recognizes the whole conformation of PBS1 rather than the specific cleavage sequence in its activation loop. And then again, the RPS5 knockout background that shouldn't be able to mount an immune response to AvrPphB shows a level of immune response that's similar to the empty vector as is expected. So this was a really great result for the lab and for me, I guess, as it showed that PBS1 decoys could confer complete immunity to a viral pathogen vectored either by Agrobacterium or by aphids, whilst retaining its original immunity to Pseudomonas syringae. But again, for this to be truly useful, we need to be able to translate it to crop plants. And we wanted to know if we could translate it to soybean using native soybean proteins. So this is work previously published by a previous graduate student in the lab, Matt Helm, but he was able to show that PBS1 is conserved in soybean. Soybean actually has 3 PBS1 orthologs. And knowing this, he wanted to test whether or not they were cleaved by AvrPphB in the same manner. So this is Arabidopsis PBS1 that in the presence of AvrPphB shows a cleavage product, but not in the presence of AvrPphB with the C98S mutation, as this is an inactivating mutation for AvrPphB. And sort of great news for us, this is the same in all 3 of the soybean orthologs. They produce a cleavage product in the presence of AvrPphB. And so for the rest of the work, we're just going to be using PBS1-1, the first ortholog. Yeah, so we wanted to test whether this PBS1 that is cleaved in soybean, whether it is guarded in a similar way to Arabidopsis PBS1. Because whilst PBS1 is conserved in soybean, RPS5 is not. So to do this, we chose to do a protoplast-based experiment in which these protoplasts are expressing luciferase along with the PBS1 protein, and the level of light that is produced can be taken as a proxy for the number of living cells. Therefore, if the protein that you're expressing in those protoplasts causes an HR, it will produce a decrease in light that you can then quantify. So I'll bring back this diagram because of Arabidopsis PBS1 to explain that this cleavage loop is actually really sensitive to perturbation, so much so that the introduction of 5 alanine residues into the cleavage loop produces a conformational shift that's recognized in Arabidopsis in exactly the same way as cleavage. So when we make this 5 alanine insertion of the soybean PBS1, that is also recognized by the plant as it being cleaved. as shown by this drop in luminescence compared to the soybean PBS1 without this 5-alanine insertion. And this really suggests that soybean PBS1 is monitored for damage in the same way that AvrPphB, in the same way that Arabidopsis soybean is, or Arabidopsis PBS1 is, sorry. So this was the good news that we needed to actually go ahead and make the stable lines in soybean. And so we made 35S soybean PBS1 stable lines, but because tenant mosaic virus doesn't infect soybean, we needed a different mosaic virus. And fortunately, there is a soybean mosaic virus that is a close relative of the tenant mosaic virus and also employs an Nla protease as part of its infection cycle. And so in exactly the same way that we made the PBS1 TUMV mutation in the PBS1 cleavage site or cleavage loop, we made the soybean PBS1 have a mutation in the cleavage loop that allows it to recognize the SMV Nla protease. And then these lines were challenged with the soybean mosaic virus, and sort of great news, what happens in Arabidopsis also happens in soybean. So that in family A and B that are expressing the PBS1 SMV transgene, we don't see any buildup of soybean mosaic virus coat protein, and we also don't see any virally derived GFP. However, in family C, in which the transgene has segregated out, you can see both visually that there is virally derived GFP here, as well as in many lines, there was virally derived coat protein. And so we also wanted to test whether AvrPphB recognition is still retained in these lines. And this was done by graduate student Alexandra Margetts in the INIS lab. And so these lines were challenged with AvrPphB soybean, with Pseudomonas expressing AvrPphB, the inactive version of AvrPphB, an empty vector or just a magnesium chloride control. And you can see that in the presence of AvrPphB, we see a cell death phenotype that's best visualized under UV light as those white dead cells. And so this was great news because it meant that just like in Arabidopsis, in soybean, the PBS1 decoy was able to confer complete immunity to a viral pathogen whilst retaining that original immunity to Pseudomonas syringae. So the takeaways from the talk and from the paper are that RPS5 requires a plasma membrane to signal, that PBS1 decoys can confer immunity to viruses and bacteria, and that this immunity is additive, it's not switched, and that it's translatable to crops using native PBS1 proteins. So our lab and our collaborators have shown that both barley and wheat are able to recognize AvrPphB. And because PBS1 is so widely conserved amongst flowering plants, and because we've shown AvrPphB recognition in both monocots and dicots, it is not insane to hope that AvrPphB recognition would also be quite widely conserved. We've also shown that like fungi, oomycetes, and nematodes all deploy effectors into the plant cell as part of their infection cycle, which makes it tempting to speculate that maybe all of these have proteases that could be targeted by PBS1 decoys. And with that, I'd like to thank our collaborators, Claire Castile and Aurélie Bach, for their aphid expertise, Dr. Jean-Francois Laliberte for providing the TUMV 6K2 GFP construct, and the Indiana University Light Microscopy Corps for access to their confocals. I want to thank especially my PI, Roger, and lab members past and present that contributed to this work. I tried to shout them out at the time, but Matt Helm, Ali Margetts, and Luke Tang are all co-authors. The work was supported by an NSF Organismal Systems grant to Roger, and I myself am supported by the Carlos Miller Fellowship from Indiana University. And finally, I'd like to thank MPMI again for the award and for the chance to present today. And with that, I will take any questions.
Sarah, terrific talk. Thank you so much. Really lovely to have you go through it. One of the things I really liked about the paper was that whole logical progression that you presented as like, if this, then that, all the way through. And that made it really interesting to read and also to follow how you did it, more understandable too. So, okay. Welcome back, Morgan, from all your live tweeting. I hope you guys, some of you will check out the MPMI Journal Twitter feed today to pick up on some of the details maybe you missed. And of course, if you have missed more of it, we will be posting this recorded seminar and people can watch it whether or not they've registered. So I hope you'll let friends and colleagues know. So going to start off with some questions. I'll take the first one. Ryan Kessens, and I should say, if you have questions, please type them into the Q&A box. So Ryan Kessens asks, do you believe that decoy engineering can be expanded to other well-characterized effector substrates?
I think it would depend on if it is similar to PBS1. I think the question is like, could other proteins than PBS1 be used? Yeah, I think if the guarding mechanism is similar, so that it is just a conformational change rather than actively the cleavage itself or the specific sequence of the cleaved ends, then I think, yes, you could probably use this in, for other substrates.
I have a question to add in on my own. So you've been doing the work in viruses mostly. Is that an intentional choice because you see this as particularly useful for antiviral immunity, or is that just because of the ease of infecting with viruses? I'm just curious about that background.
Yeah, so mostly I think through the ease of being able to infect with viruses, especially the Agrobacterium-based infection that we had is very easy. But in the lab now, we're currently looking at Ali Morgatz is doing stuff with nematodes and with their infection and their effector process.
Cool. Yeah, I can imagine though with the viruses, because those proteases are so important for their lifestyle that it's maybe more useful with antiviral than like with an antifungal where a protease, there's one of 100 or more effectors. So I was just thinking about that. Yeah,
the NA also provides like a really solid sort of not easily evolved around recognition mechanism.
Actually, that kind of brought up a question to me thinking about, one of our other top 10 questions is why do some species have so many effectors and some have so few? And you're right that with viruses, I mean, they've got limited genomic space, right? And so that one protease is not going to change. But I'm curious, I wonder for some of the ones like nematodes where they can be secreting in a number of effectors, whether this, because it's so good at activating that defense response, whether this could be a way, I would think that it would be very valuable as long as you can find one protease in there that is still, that's required, that's still working, even if you don't know the cellular target.
Yeah.
That could be good.
Yeah, and I think so long as you're not sort of bringing out a whole field, like hundreds of fields, all expressing the same PBS1 decoy, and you had more of a patchwork, hopefully the evolution pressure would not be as huge.
Yeah.
So are there other like crops that y'all are most interested in switching to, or where do you see this going next?
So my project is moving, or like my focus is moving more into like finding maybe partners for RPS5. And also it would be really useful for us if we had a structure for PBS1, because as I said, the cleavage loop is like pretty sensitive to perturbation. And so some of the cleavage site sequences that we tried to put in there have actually caused autoactivity. And this was like a big issue for like Matt Helm, that he would do like a month of cloning just to find out that, sorry, that one doesn't work. And so it would be really useful for the lab to be able to have a PBS1 structure and more intelligently be able to design what cleavage sequence we could actually use and which ones are not. And so this is something I have been trying to do, but it is proving not super simple, but it's also been a huge learning curve for me to learn to do protein expression.
Yeah, I remember meetings with Matt where he was commiserating about that. So
I think it happened to him like 5-6 times.
So we have a question from.
So we have another
go ahead,
question from the Q&A, which is by Andy Reed. Great talk. Do you have a library of endogenous plant proteases and cut sites to stay away from when you're engineering to prevent autoimmunity?
I wish we did. No. So currently in the lab, we're working on a way to be able to screen proteases to find what their cleavage site sequence is. And that would be a really useful tool to be able to get up and running. But no, at the moment, it's completely trial and error. So yeah, that was a real issue that Matt Helm ran into multiple times. And I guess once we know it doesn't work, we're not going to do it again. But other than that, there's no library.
I was really intrigued by the fact that the RPS5 can only work when it's at the plasma membrane. And I know you're searching for interacting partners. I wonder, do you have any candidates in mind, or are you really hoping that this will open up, that you'll find something unexpected? What's your thoughts?
Yeah, I'm sort of hoping that there'll be something unexpected. Because, well, mostly I was hoping that it would function similarly to ZAR1, because that would be really exciting for us. But as it seems like apparently there's really very different ways that CC-NLRs are doing their signaling. Yeah, I think we don't really have any candidates for what might be associating with RPS5. It's sort of nice that we have PBS1 as our positive control. So if we don't see that, the lab has previously tried just doing like mass spec, like IP mass spec, and have not seen PBS1 come down. And so it's the PBS1 we're going to work in as a sort of inbuilt positive control, as we know they associate via co-IP. Yeah.
So I don't think we have any more questions in the Q&A box, but just to remind people to type them in if you have questions. But I guess I was thinking, since you are the graduate student best paper award winner, whether you might give any tips. You wrote a terrific paper. Obviously you have a great project, but any suggestions for graduate students writing their first papers?
Gosh, I don't know. Whenever a project gets to, and Roger was really encouraging of this, when a project gets to a point where you think there's enough data that like somewhat of a story is developing, I find it really useful to make figures, write the figure legends as soon as you have done that experiment, so that you're not having to check back through your lab notebook to be like, what was the incubation time on this? I don't know. And then once you have all of those figures, you can sort of plan ideally the next figure that you want, and it's easier to see if there's like logical missteps. But yeah, other than that, I found the review process very, very helpful. We got the paper changed quite a lot in between the first and second review, and I think it was made a lot stronger. Yeah, I had my mom read it, I had my dad read it, I had Roger read it. If you have non-science people reading your paper, I think it is a lot easier for them to see where the logic just doesn't make sense. Yeah.
Interesting, good advice. There's so many different ways to write papers. That's why I was curious. But I agree, when you start mocking up the figures and putting them up there, sometimes you realize that you really needed one more little panel, right, to complete that bit of logic. And sometimes you can't really see that until you put it all together. So totally. And I think that whole getting other people to read it who are not in your lab is key, because that's how you write an understandable paper. Also, I'm delighted to hear that the peer review process went well. That's something as a journal that we really aim for. We'd like, I also feel that my papers have been improved by comments from people who were not in my lab, really helpful. And I see here from Kamal, sorry, I'm having a little trouble here with my chat. Kamal Malukani has posted in the chat, he said that last advice is a gem. Ask non-science people to read it. Thanks for that. So, okay, I think we are going to stop here. So thank you so much, Sarah, for your wonderful talk, and looking forward to it. to seeing the recording up. Yes, congratulations. And Morgan, thanks for co-hosting. So just to remind you that that recording will be up soon. You do not need to register for it. It will be up soon on the website where you registered. And I think, Ashley, if you could just paste that into the chat so people can easily find where to come back to see the recording. And thanks to those of you who have volunteered to help edit the transcript to make this more accessible for our colleagues. And that's a real push we're trying to do now, is to increase inclusion in that way, to make our talks more accessible and our tweets. Morgan has been putting all text into all her images to make them more easily understandable by a screen reader. So if you still would like to volunteer, please let me know, and we'll happily include you in that process. Okay, well, thanks to everyone. Thanks especially to Sarah for a very lovely talk. And we will see you at our next What's New in MPMI. Bye, everybody.